Welcome one and all for this continued lectures of paper 8 of biotechnology, which is industrial biotechnology and environmental biotechnology. So in the previous two sessions, we have discussed about what is fermentation technology and what is bioprocess technology. What are the differences between fermentation and bioprocess? Differences between a fermenter and a bioreactor. We have also discussed about the types of the bioreactors. We have also discussed the raw material or the media substrates that are probably used for this fermentation process, and we have also discussed about solid state fermentation process. Now in today's class we will be discussing about an overview of fermentation process, and we will be discussing the types of fermentation process. So this is the overview of fermentation process. It begins with the stock culture and then inoculum development, and then we transfer it into the seed fermenter, and then it is sent into the bioreactor. So these are the events that are going to take place in the upstream process. So till here it is upstream process, and this is your downstream process. We call it as DSP, and this is USP, right? This is the overall fermentation process, and the downstream process we will be discussing separately, and in detail we will be discussing DSP, right? So now we'll be discussing the overview process of fermentation. Yeah, this is another way of representation of the bioprocess. Here it is clearly mentioned what is upstream and what is downstream. So this is your downstream and the events that are going to take place in the downstream, and these are the upstream processes. So where upstream is mainly involved in the development of a inoculum and uh, putting it into the bioreactor, which is containing a suitable raw material. And downstream is all about the separation, to collect the harvest, separation, purification and finishing. All these steps are involved in the downstream processing, right? So the fermentation process basically consists of inoculum preservation, inoculum buildup, pre-fermenter culture, and finally production fermenter. A brief account of that is being shown in the diagram. Uh, the first step, the inoculum preservation. Inoculum is nothing but so inoculum is our microorganisms, or the organisms or the cells that we use. They are called as inoculum. So inoculum is nothing but our organism of our interest. So organism of our interest, we call it as an inoculum, right? uh, it has to be preserved and it has to be maintained. See, in strain development we have discussed how an industrially important strain can be isolated, and we can preserve it. To preserve, there are many technologies, there are many techniques by which we can preserve. We can store in the refrigerator, right? We can store by freezing. We can preserve by the organism of interest by lyophilization. So once the organism is preserved and we collect that cell or the organism, an inoculum has to be built up. The actual process and the condition for the inoculum buildup depend on the preservation technique used. There are wide variations in the growth times which depend on the type of the preservation. So in refrigeration, we require around 2 to 6 degrees centigrade, so which is suitable for bacteria, actinomycetes and fungi, and they require different growth time. So bacteria doubles in 6 to 24. Actinomycetes, they take about 1 to 3 days. Fungi, they require 1 to 5 days or 7 days. So frozen culture, bacteria, lyophilized cultures, yeah, this is the same thing it is mentioned here. So frozen cultures, bacteria require 6 to 48 hours. Actinomycetes it is, please mind this mistake. It is actinomycetes 1 to 5 days, fungi 1 to 7 days for all organisms. And yeah, just let me make this correction here. Yeah, so it is actually actinomycetes, right? So it is not there is a spell mistake, it is actinomycetes. And fungi, it requires 1 to 7 days. Lyophilized, for all organisms it requires around 4 to 10 days. Pre-fermenter culture. Pre-fermenter culture or pre-culture or pre-fermenter culture is often required for inoculation large sized bioreactors. Inadequate quantity of inoculum will not only delay the product formation but also reduce the yield drastically. So there should be some proportion between the raw material and the so if you have a vessel which is containing around 1 liter of your raw material, so according to the mass of the raw material, the inoculum will be, so usually the inoculum will be around 1% or less than 1%. So if your volume is increased from 1 liter to 1000 liter, so your inoculum also has to be increased. So it has to be 1%. So from 1 ml it has to become 10 ml. And if it is in large bioreactors, if the volume is in gallons, so even the inoculum has to be increased. It has to be proportionate, right? So if we just put a little bit of inaccurate inoculum, it may certainly take large time for the formation of the product and it may reduce the yield. So that is why we require pre-fermenter culture. Production fermenter. For appropriate production by fermentation process, several parameters need to be carefully considered and optimized. These include composition of the medium, carbon, percentage of carbon, nitrogen source, batch to batch variations, effect of sterilization on the nutrients and pH, and alterations in temperature and aeration. So these are the important parameters that has to be kept in mind while doing large scale fermentation process. So again, this is a diagram, and now we will move on to the types of fermentation process. The fermentation process, it can be either basically we have three types of fermentation processes. Number one, it is batch fermentation, and we have fed batch fermentation, and we also have continuous fermentation. So a batch fermentation is regarded as a closed system. The sterile nutrient medium in the bioreactor is inoculated with microorganisms. The parameters are all under optimal conditions. It follows 6 typical phases of growth are observed in the batch fermentation. So we have we can expect lag phase, log phase, stationary phase, and a decline phase, right? So we can expect lag phase, log phase, stationary phase, and a decline phase. We can expect in a typical batch culture, we can expect this lag phase, log phase, stationary phase, and a decline phase we can see in the typical batch fermentation process. And it is a closed system, remember this, because you can't add either media or you can't add the microorganisms, nor you can remove the product. So it is for one batch. So the volume is limited, the space is limited, the raw materials are limited in case of batch fermentation process. So in lag phase, so I have shown you here initially this is the phase where the inoculum is being put into the reactor or into the fermenter. The microorganism adapt to the new condition, new atmospheric, new environment, and here there is no active division of the cells. So when we enter into the acceleration phase, uh, this phase connects to the lag phase, and actually lag phase is the phase where you can see there is a increase, steep increase in the, there is an increase in the microorganisms. Doubling of the microorganisms is more active during the log phase, right? Deceleration phase, as this growth rate of microorganisms during log phase decreases, they enter the deceleration phase. This phase is usually very very short, and it is close to the stationary phase. And in stationary phase, as the substrate in the medium gets depleted, the cells stop dividing and the cells enter the stationary phase. The microbial growth may either slow down or completely stop. And the last phase in the batch culture is death phase. The phase is associated with cessation of the metabolic activity and depletion of the food reserves. The cells die at an exponential rate. So this is the growth curve, and this is the growth dynamics that we can see in a batch culture. Fed batch culture, it is an improvement of batch fermentation, wherein the substrate is added in increments at different times throughout the course of fermentation process. In batch culture method, substrate is added only at the beginning of the fermentation. So in case of batch culture, no further additions. Uh, once you put all the things, once you put your raw material, once you put your inoculum, it's close, it's over. So the cells will start, the cells will go into the lag, log, and then stationary, and then decline. Whereas in case of fed batch, in between, as soon as the cells try to enter the stationary phase, fresh media is being added so as to prolong, the log phase, the log phase is prolonged here in case of fed batch culture. The major limitation of fed batch fermentation is that the microorganisms in the stationary phase produce proteases. These enzymes attack the recombinant proteins that are being produced by the cells. So hence it has to be properly monitored. Third one is continuous culture or continuous fermentation. So continuous fermentation is an open system. It involves the removal of culture medium continuously and replacement this with a fresh sterile medium in a bioreactor. So there are two types of continuous fermentation, and bioreactors are described below. Homogeneously mixed bioreactors and plug flow bioreactors. Yeah, this is the comparative statement of the different type of growth patterns that we can see in batch, fed batch, and continuous culture. So if you look at the concentration of the cells versus the time, see, when I come to batch culture, the cells are increasing, right? And the cells enter lag to they enter, yeah, let me use some colors. Yeah, see, the cells are entering into lag phase, log phase, and the stationary phase, right? And they come down to depletion phase. And when you look at the concentration of the substrate, I will use a different color. And when you look at the concentration of the substrate is coming down. The substrate concentration is coming down. So till there is a substrate concentration is high, the cells are dividing actively. When as soon as the substrate starts to deplete, the cells will enter stationary phase. So this is how we can see in case of batch fermentation. So when I come to the fed batch fermentation, so in fed batch fermentation, yeah, the cells are increasing till there is a substrate concentration. They are entering the log phase, and then they are near to the stationary phase. Immediately fresh culture media is being added. So when you add the culture media continuously, it prolongs the stationary phase. It prolongs the stationary phase. The stationary phase is being prolonged. The stationary phase is being prolonged. So in case of fed batch culture. And in case of continuous culture, you see the concentration of the substrate is linear and the concentration of the cells is also linear. So here the substrate is continuously added, the products are being continuously removed. So it is something like parallels and there are no peaks. Yeah, again batch versus continuous fermentation process. When you just look here, yeah, when you just look here, this is a closed system. So once you are putting the raw material in and the system is closed and your raw material or your substrate is converted into useful product and you remove that product. That's the story. And the microorganisms, they behave lag phase, log phase, stationary phase and then decline phase. And in case of a continuous fermenter, the feed, there is an option to supply the feed continuously and you can remove the effluent or the waste material. Effluent is the waste material. The waste material can also be removed continuously. So this is the fundamental difference between the batch versus the